Time for some fun, guys. Let's look at fungi. Be able to define and identify the specialized adaptations of fungi, including mycelium, spore, heterokaryotic, and hyphae. Also zygospore, ascospore, and basidiospore. List traits specific to the chytridiomycota, glomeromycota, ascomycota, and basidiomycota. Relate surface to volume ratios to diffusion rates. Draw the life cycle of a fungus, labeling the haploid, diploid, and heterokaryotic phases. For every one of those terms, label where it occurs in the life cycle. There's your pun. Fungi evolved from the opisthokonts, so they are pretty similar to those amoebas, remember? But they're more closely related to animals than plants, even though they were incorporated in botany in the past. Their ancestor was a protist, and, well, fungi, animals, and plants. Their ancestors was all protists. It's a paraphyletic group. So let's look at the phyla of the fungi today. We're going to look at the chytridiomycota, the glomeromycota, and the zygomycota within that, the ascomycota, and the basidiomycota. It gets more fun as we go along, so hang in tight. But first, let's look over a little physiology. What do fungi do? Well, fungi aren't just a mushroom. There's much more to fungi than that. Fungi are, first and foremost, heterotrophic. They need to obtain nutrients from autotrophs, but they can also obtain nutrients from debris. This makes them called saprobic. That's the ability to actually get the nutrients from rotting matter. Some fungi are parasitic and some are mutualistic. Fungi are primarily terrestrial. We're going to look at chytridiomycota. They are aquatic, but everything else you're going to find on land. And they run into terrestrial problems, of course, like drying out and UV light. Wait, not UV light, because they stay underground. The mycelium is a vast network of filaments that grows underground, often unseen. Although here in Washington, if it's wet enough outside, you can kick some leaves over and see all this white mat. That's mycelium. And fungi reproduce using spores. And spores, if you remember, are haploid, and that means the fungus, for most of its life, most fungi are haploid. And this mycelium allows them to have a large surface area to volume ratio. So exchange occurs across surfaces. Think about the alveoli in your lungs or the villi in your intestines. These are surfaces across which exchange occurs. And fungi use their mycelium to absorb things from the environment. The fungal mycelium is huge. It can grow up to 1 kilometer per day, but not 1 kilometer in a single direction. No, we're talking about all the different branches that can grow in a cubic meter of soil, can be up to 1 kilometer. It's a huge amount of growth, and it's a real large amount of surface area. If you're thinking about what a fungus looks like, it's not a mushroom. It's a large net underground. And we're going to see that surface area to volume ratio come up a few more times in biology, so here's a good a place as any to start it. How do fungi do what they do? Well, their ecological role is in decomposition, as you see by that yummy strawberry. Not yummy to you, yummy to the fungus. They have roles in decomposition and nutrient cycling. They're an essential part of an ecosystem. Yes, bacteria can also decompose, but you can't decompose as well as a fungus can. Lignin does not get degraded easily. If you remember those calamites and lycopods that fell in the swamps and didn't degrade, but rather turned to coal, that's because fungi weren't there to eat them. So lichens also secrete lichen acid, which can dissolve rocks. And this dissolving rock is essential to turn rocks into soil, which can later support plants. So fungi are having a very essential role in any ecosystem. We're going to start our survey of fungal phyla with a truly nasty one, the chytridiomycota. These are often parasitic. One of the unique things about these guys is that their spores have a flagellum, and they are aquatic, which is why having a flagellum is useful. You may be familiar with Botrytio, and that's going to be your frog-killing fungus that is currently leading to a decline in frog species worldwide. It parasitizes the frog's skin, making it hard for the frogs to breathe underwater, and they die. It's a terrible plague, and it is a chytrid fungus plague. These are not very different from the protists that they evolved from. You can actually look at the nearby protist groups and see that they also have flagella and they are also aquatic. But moving on to the land, we see the zygomycota and glomeromycota. The zygomycota is what the group used to be called, and you might see this in some of the older books, but the zygomycota are now part of the glomeromycota. These have a mycelium that grows in the land or can grow in your bread. They're a decomposer, and when two different mycelia meet, they fuse, and these two haploid organisms are going to fuse and make a diploid zygote. That is the zygospore, because upon becoming a zygote, the spore immediately undergoes meiosis and makes 4 more spores. So this is a good way of reproduction and a good way of making new spores. The spores lack flagella, which is why you'll see them on the outside of the breads. So they do not wait long after meeting 2 mycelia to make a new spore. The glomeromycota were once a smaller group in the zygomycota, but have now become their own group, even subsuming the zygomycota, as we realize just how many species of mycorrhizae there are in the world. These are the fungi that form arbuscular mycorrhizae with plant roots. You can see those little balls in that plant root. That's the fungus living in the plant root, exchanging nutrients with the plants for carbon. And this is essential for the wood wide web, which connects all the trees in the forest with mycelium, thus allowing the trees to share nutrients with the mycelium and even with each other, and allows trees to even communicate with one another. 80% of plants partner with glomeromycota fungi, but it's very hard to tell which species is which because they don't make mushrooms and they don't make really visible things, so you need genetic studies to determine what species are which. The ascomycota are the sac fungi. These have cup-shaped fruiting bodies. As you can see with this pizziza here, it looks like a little cup, and a raindrop will fall in that cup and disturb a whole bunch of spores, launching them into the air. These are formed on the ascocarp, which is a special fruiting body. You can see a morel here, and you can see a truffle down there. Those are different ascocarps, and the type of spores are called ascospores, and they are found in an ascus. Ascus just means sac. These are sac fungi because you put 8 ascospores in a sac. Well, how do they form these spores? That's meiosis. So meiosis followed by 1 round of mitosis is what's needed to make all 8 of those spores in each one of those sacs. Well, how do they become diploid? Well, 2 mycelia meet and they begin to form the ascocarp, which is not entirely going to turn into spores. We're going to get back to the terms of that later on. But the whole thing doesn't turn to spores. Rather, it waits a little while, forms an ascocarp when conditions are right, and then forms ascospores. This delay in reproduction is adaptive because it allows more spores to be made from a single reproductive event. And this makes them common worldwide. We see a lot of these species. A lot of lichens are actually working with the ascomycota and an algal species. We're going to look at those in a later lecture. The basidiomycota, welcome to the club. These don't make sacs, they make tiny clubs. And those tiny clubs are going to be on the bottom of a basidiocarp or a mushroom. So the mushroom is a basidiocarp, and these are the only true mushrooms are the basidiomycota. There are also shelf fungi, which are in the basidiomycota, and underneath those shells are tiny gills, and on those gills are tiny clubs, and on those clubs are tiny spores. Those are basidiospores. And it's not like the zygomycota where 2 mycelia will meet and immediately form a mushroom. No, 2 mycelia meet and they actually share their nuclei to do something called a heterokaryotic phase. More on that later. It delays reproduction for a long time. In fact, you'll see these being able to reproduce multiple times from a single meeting. You'll see these fairy rings in your lawn. That's the heterokaryotic phase spreading out and forming mushrooms on the outside of the spreading heterokaryotic phase, allowing these fungi to reproduce even more from a single meeting of 2 mycelia. They can make billions of spores, making them a very successful fungal phylum. They are decomposers, some are mutualists, and let's face it, some of them are delicious and they're food sources, and some of them are deadly, and some of them will make you trip. So the reproductive anatomy of these, let's look at some of these terms. We look at holocarpic and eucarpic. You remember those zygomycota? They're holocarpic. When 2 mycelia meet and fuse, the entire structure becomes reproductive. So the entire structure being reproductive is holocarpic. Eucarpic means not the entire structure becomes reproductive. And that's going to be true of your morels. Most of that is not spores. The ascomycota have a large vegetative structure that is not reproductive. And the basidiomycota, even more so, having a vast mycelium that is non-reproductive. There's the diaspore involved in mitosis. So this is one of these spores that forms by mitosis, so they can make spores without needing reproduction. Spores themselves are often the products of meiosis. And we can look later at sporangia versus conidia if you want to really get into it for how these fungi reproduce and look at yeasts and stuff. It's kind of interesting. You can really go pretty deep with these fungal reproduction. But we're going to look mainly at this phase. We're going to look at a few terms here. You have your germination of a spore and the formation of a mycelia. Now I said sometimes a mycelia can make its own spores, and it can do that through asexual reproduction. Those are going to be called your diaspores. Mycelium can just undergo mitosis and specialize that to make more spores. That's asexual. That's great. You germinate those, you make more mycelium. Eventually, two mycelium are going to meet, and they're going to fuse their cytoplasms in what's called plasmogamy. Plasm meaning cytoplasm, and gamy meaning marriage. So they marry their cytoplasms, but they don't unite their nuclei like you'll see in the fertilization in animals. Rather, the two nuclei stay distinct, and those distinct nuclei existing within cells are heterokaryotic. Remember, karyon meaning nuclei, the eukaryotes and prokaryotes. These are heterokaryotic because they have two separate and distinct nuclei. Eventually, those nuclei will fuse in karyogamy. And when they do, they can form a zygote, and that zygote will undergo meiosis, giving rise to spores which can begin the cycle anew.